Okay.
Okay.
For you.
All right. So thank you guys for coming out on this like kind of dreary, gross Sunday. This is our last final exam review. So how are we feeling so far? Or you can just give me thumbs up, thumbs down.
I feel like I quite honestly the best in innings. I'm doing it Tuesday. Not for any medical reason, I just want more studying time.
I don't know if
I don't think that will work.
yeah. Um I feel three also. Okay. So yeah, our final is tomorrow. Make sure you're in the right room. Make sure you know where it is before it's like time.
Wait.
Oh, and it is not in Moral 2 131. That is from last semester. So do not show up to Moral 2 131. I'm pretty sure it's in, is it in Boyden Gym?
Yeah, they still Boyden Gym.
Okay.
God, I hate that goes in that's in Boyden Gym.
No, it should just be the Google Doc, but if you're worried about it, I would send Laura an email and just make sure that she's gotten that. Yeah, do not show up to Moral 2 131, please. The exam time and like location is on Moodle. All right, so we're gonna start off with cell cycle 2. We got split up between like our last topic, cell cycle 1. So we're gonna finish that off. We're gonna start with S phase because G1 was a part of our cell cycle 1 unit. So first we are gonna start off with pre-ICs. So on the DNA strand, when we're gonna replicate our DNA, right, we have already formed our pre-RC, our pre-replication complex, where we have those first couple of proteins building, getting ready to form the replisome, getting ready for DNA replication. So after we have our pre-RCs, that's basically a scaffold for the pre-IC. There's not a huge difference between the pre-RC and pre-IC. The pre-IC basically just happens later and it has more proteins added onto that complex. Pre-RCs can form when there are pretty low levels of active CDK to like phosphorylate substrate. We just don't need that much. But pre-ICs form later because we need more active CDK, probably because more of those proteins that we're recruiting need to be phosphorylated to add on to that like complex at the origin. So after our pre-IC forms, that kind of structure is what recruits DNA polymerase to the origin. And that whole big complex together is called the replisome. So when we start synthesis from an origin of replication, like with one of the replisomes, that is called an origin firing. And when we start firing our origins, when our origins start replicating, when our replisomes become active, that means that we are in S phase. But again, like just based on the amount of time that it takes to phosphorylate all of our substrates and recruit DNA polymerase, we have a lot of origins of replication along the DNA strand, and they won't all fire at the same time. We just say that we're in S phase once we have like our initial firing happening. And then once we've fired at our origin, that basically means just starting to replicate at that point. So once we've fired at an origin, we want to suppress re-replication or refiring at that spot. Does anybody know why that is? You can just, yeah, go ahead.
So you get an extra replication?
Yeah, exactly. So basically, like once our origin has fired, we don't want it to do it again because we could end up with broken DNA, we could end up with too much DNA, it would just be an error in replication. So we have a lot of mechanisms that are going to prevent re-replication from
I
happening.
remember that the last thing you mentioned, the topic notes mentioned that it would basically make the cells unhappy, basically when it's an overbuilding of DNA, that's it's building DNA and like the protein in general, just gone under crap that they with the campus.
Yeah, exactly. Like once we refire, we're opening up a whole bunch of problems that could potentially happen. Anytime that we have like problems with our DNA, that could lead to cell death, it could lead to overproliferation, it could lead to broken chromosomes. So the cell has a lot of somewhat redundant mechanisms to prevent refiring because we do not want our origins to refire. So our first mechanism that we have here is CDK phosphorylation of ORC. Just in case you don't remember, ORC is the origin recognition complex. It's a protein. And when CDK phosphorylates ORC, CD6 binding is prevented. So that phosphate group that we're putting on ORC, it basically just blocks the binding site for CDC6. Our next mechanism that we have is CDK phosphorylation of CDC6, and that basically flags it. So a ubiquitin ligase, which is a type of enzyme, knows to put ubiquitin on it. And when we have ubiquitin on a protein, it sends it to the proteasome. It's like a signal to bring it to the proteasome for it to be degraded. So we can consider like ORC inactive or it can't be bound. CDC6 gets degraded. And then our last mechanism is CDK phosphorylation of the MCM2 helicase complex. And that just exports it from the nucleus. So we have one that can't be bound, we have one that's degraded, and we have one that can't be accessed because it's in a different physical location. Go ahead.
This confused me on the stopping us. What are those diamond structures in the S phase? When CDK is activate existing free proteins.
Maybe?
Yes. I simply were to see.
I wonder, is it supposed to be phosphorylated? I don't think it matters. Like, you don't have to know what this is.
So long as you know that the replication bubble exists.
Yeah, the diagrams that we use, like, they align pretty closely with what we cover, but sometimes, like, if there's a little thing that you don't know what it is, it's probably just because we didn't talk about it, and I wouldn't worry about that stuff. So does somebody want to like kind of throw out an explanation for why we might have 3 redundant mechanisms to prevent origin refiring? Go ahead.
It's really bad when you have a lot of extra one of them failed or one of them is like another problem. That is, and has to be activated for something else you want.
Exactly, yeah. So each one of these sequential redundant mechanisms is like a safety net. So if one fails, for what, like if we have a gain of function in ORC where CDC6 can still bind it, we have 2 other mechanisms that's going to prevent rebinding. And because the cell's main priority during S phase is making sure that it has correct DNA replication and no damage, we need those fail safes. And also, CDK doesn't phosphorylate ORC, phosphorylate CDC6, phosphorylate NCM in that order. All of that is happening all at once. So say like it hasn't reached all of the ORC proteins, it might still have already phosphorylated an MCM that can't then access and add to the replosome. So these are all just like kind of contributing to make sure that we can avoid refiring. Okay, so then we have cohesin. Cohesin are these little ring-shaped proteins. They're kind of cool. And they hold sister chromatids together after they're replicated to prevent early anaphase. So until they are cleaved, our sister chromatids can't be pulled apart to either pole of the cell, and that allows us to wait until we're totally ready. And they are loaded during S phase, and that is like one of the requirements for the spindle assembly checkpoint, that cohesin rings have been loaded properly. So the S phase checkpoint, in most textbooks, you probably won't see it. It's kind of like an informal checkpoint. We call it a checkpoint because even though it doesn't have as many mechanisms behind it, before we progress from S phase to G2, we want to make sure that all of our DNA is replicated and that it is not damaged. And we can stop temporarily at this point if we sense problems to try to fix our DNA, or if we can't fix it, go through apoptosis. Okay, so G2. Is everybody okay with S phase? Okay, we can always come back. Okay, so G2 technically stands for gap, but if you want to think of it as growth, the main thing that's happening at this point is that we are making sure that like we have enough cell mass and division of proteins and organelles to divide into 2 complete daughter cells. Okay. So basically before you divide, you need to about double your size, the size of the cell. If you don't do that, then every division you're going to get smaller and smaller and smaller. So like everything that you have in the cell, the amount of cytoplasm, the amount of mitochondria, the amount of chloroplasts, your organelles, they all have to replicate, your proteins. And so that is like one of the things that's going to be happening in S phase. And this is also going to prep us for chromosome segregation and M phase. But we don't enter into M phase until like we've passed the point where like we've ensured that we have enough growth, that we don't have any DNA damage. So again, a lot of these checkpoints are redundant with each other because it is the worst thing for the cell, for the organism to have damage to DNA. So you can kind of think of all of these things as like occurring in patterns where like we're going to take a little step and then we're going to do a whole check through to make sure that we don't have DNA damage, that like everything is going as it's supposed to. So in early G2, this is where we have like that pathway with WEE1 that I like. So we're getting ready for M phase. And once we're ready for M phase, we want to be able to like activate all of our M phase CDK really, really fast and just like get going with it. So our M cyclin levels will rise during G2, but our M CDK will remain inactive. So you guys remember how cyclin and CDK work together? Does somebody want to explain that? What does CDK stand for? Yep. So cyclin and CDK bind, right? What does cyclin binding do for CDK? Go ahead. Yep. Yeah. So when we have cyclin and CDK binding, cyclin is going to cause a conformational change in CDK that basically pulls out the T loop or the activation loop. So it can be phosphorylated and then that CDK kinase will be active, have active kinase activity. And the binding interface between, so like if this is cyclin and this is CDK, they come together and then the binding interface is like this space in between my hands. So their substrate will bind both of them. So the cyclin gives the CDK specificity for its substrate, if that makes sense. Go ahead.
Is the WEE1 inhibition only in the G phases, or is that in every?
In the scope of what we learn in this class, we are only thinking of this as being in G2. So
no WEE1 and G1.
No. So yeah, we have our increase in M cyclin production. So we're getting ready for M phase after G2. We have MCDK and M cyclin bind, and then we have an activating and an inhibitory phosphate. So our inhibitory phosphate is WEE1. Usually we see phosphates as being like an on switch, but in this case, the phosphate that WEE1 kinase puts on the CDK holds it inactive. And at the same time, we have CAC, CDK activating kinase, put on an activating phosphate. So we have our cyclin CDK complex, we have CDK phosphorylated twice, and then for it to be activated, all we have to do is take off the inhibitory kinase from WEE1. So we're basically getting everything ready to go. And then like once you take off that kinase, it's like, you know, off to the races, it's ready to go. Yeah, I think that's all for this slide. Yeah. So after we pass the G2 checkpoint and we've ensured that like we have enough growth, that our DNA all looks good, then we have CDC25, which was a phosphatase. Does somebody want to say what a phosphatase is? Exactly, yeah. So phosphatase is like the opposite of a kinase. It's just going to be taking off a phosphate. So we have CDC25 activated by a protein called PLK1. It's a kinase, so it phosphorylates CDC25 at low levels. So low amounts of CDC25 are now active. CDC25, because it's a phosphatase, takes off the kinase on CDC that was put there by WEE1. So it no longer has that inhibitory phosphate. And this is where we have that positive feedback loop. So once we take off by CDC25 our inhibitory phosphate, then our active complex with the activating phosphate on this T loop that we got from CAC is gonna go back up and it's gonna phosphorylate more of CDC25 phosphatase. And CDC25 phosphatase is gonna take off more and more inhibitory phosphates from that complex. We're gonna have more activated, and then that's like kind of how this pathway increases and amplifies itself through that feedback loop. Does that make sense to everybody? Do we have any questions on this?
Yeah,
Oh, yeah, go ahead.
on that last point,
Mhm.
it says, so is it CDK phosphorylating we want, so that inactivates it?
Where did we say that? Oh, yeah, I forgot that. So no, that is gonna be, yeah, it is, right? Yes, MCDK, yeah. I always think of the positive feedback loop, but we also have a negative feedback loop, where our MCDK, once it's activated, is also going to downregulate V1 by phosphorylating it. Thank you for reminding me of that.
This is still all in G2, right?
Yep. Yeah, we're getting MCDK ready during G2 to like go during M phase. Which once
G2 CDKs or?
Yep. Yeah, and those will be active during G2. During G2 we have active G2 MCDKs, and we're making MCDKs also during G2, so we have them ready for the next phase. So once we have that active complex, we'll basically be in M phase. So we're gonna send these slides out. I'm not gonna go through like every little bit of this. It's just cell cycle. The important thing I think is just that you kind of like know what all these phases are. So, you know, in prophase, we have chromosome condensation. Prometaphase, we are like looking for our kinetochore attachments. So that's when we have that dynamic instability with our microtubules where they're reaching out and then pulling back to try to grab onto a kinetochore. Metaphase is where we line up our chromosomes on our spindles, and we have the spindle assembly checkpoint, which we will talk a lot more about. Anaphase is chromosome segregation, and telophase and cytokinesis is when we form our two daughter cells. So prophase, you don't need to know a ton about prophase. Basically, you just need to know that lamins and cohesin both get phosphorylated. And when we phosphorylate lamins, our nuclear envelope will break down, and that will release our genetic material. And because we have phosphorylated condensin, we will also have fully condensed chromosomes instead of just like that spaghetti-like chromatid that we have otherwise. And we have the very, very beginnings of spindle assembly, which is done with maps and motors, and we'll talk about that more later too. Oh, we're gonna email these out, but I can go back to it for a second.
Actually taking all less pictures.
Okay, so spindle assembly. The beginnings of spindle assembly are during prophase. For this, I would know the difference between the three types of microtubules. So we have aster microtubules, interpolar microtubules, and kinetochore microtubules. I think kinetochore microtubules are probably the most intuitive. They are extending from our spindle poles on either side of the cell, and they're grabbing at the kinetochore of the sister chromatid. So those are going to be the ones that are eventually shortening and pulling the chromosomes to either spindle pole during anaphase. Interpolar microtubules are overlapping in the middle. So these are the ones that egg 5 will sit on, which is one of our kinesin 5 family proteins. And again, during anaphase, like that will help us push our spindle poles apart, and we'll talk a little bit more about the push-pull model. And aster microtubules attached to the cortex of the cell membrane, so like the inner surface, and those are structural. They'll also contribute to push-pull. Their name seems a little bit random compared to kinetochore and interpolar. But the way that I remember it is these are the green ones that are coming out here, kind of like it looks like flower petals, and an aster is a flower. So if that helps anybody, I think that's why they named them like that. Yeah. Okay, oh, go ahead.
So are they interpolar? They overlap in
They are connected to the spindle poles, yeah. And they come out and then they overlap with each other, and they have proteins bridging them. Go ahead. Yeah, they just have different rules depending on like what they're attached to will affect what they're pulling or pushing on, and they'll all have different motors and maps that are associated with them. We okay with that? So the kinetochores are a really cool protein complex that sits in the centromere of the chromosome. They look just like little buttons on there. They're the red things, but they have there are like over 80 proteins that are all working together to form this functional protein complex. They're very cool. And kinetochore microtubules are the things that are going to be reaching out from spindle poles and binding to the kinetochore. And we'll talk more about this, but when we have kinetochore microtubules bound to the kinetochores on the sister chromatids, that creates tension, like a pulling force from the spindle poles. And we're not positive about this, but some research has indicated that once we have attachments on all of our kinetochores, that pulling caused by the spindles or the kinetochore microtubules attached to the kinetochores is what signals to the cell that we have all proper attachments and that we're ready for anaphase. Okay, so building the spindle with motors. This is push-pull. So can you guys give me like a thumbs up or thumbs down middle on this? Because I feel like people either like love this or hate it. So where are we at in here? Okay, neutral, some thumbs up. Okay. So one thing that I would remember for this, going back to like unit 6, I think when we talked about microtubules, is that our microtubules are polar. They have a plus end and a minus end. And the majority of the motors that we talk about are going to be only plus end directed or only minus end directed. So it might be good on your note sheet to just like make note of, hey, these are the plus end directed motors, these are the minus end, because if you get a problem about them, like that will kind of determine if it's a pushing motion that's happening or a pulling motion, depending on what direction they're walking in. So dynein is a minus end directed motor, and it sits on the cell cortex with our aster microtubules, and it's anchored to the inside of the cell cortex, right? So dynein will sit holding an aster microtubule on the outside of the cell, and when it walks towards the minus end, it's almost like pulling in a rope because it can't move. It can't actually walk down the length of the aster microtubule because it's anchored to the cell cortex. So as it pulls towards the minus end, what that's doing is it's dragging the spindle pole towards itself. The analogy that somebody used in my office hours, like last semester, I think, that I thought was really helpful is instead of like kinesin that walks along the length of a microtubule like a road or a bridge, dynein is kind of like a person sitting on a dock and pulling in a boat by a rope from the dock. So like our boat would be the spindle pole and our rope is the aster microtubule, and then our dynein is our person. So it's not moving itself, it's moving the thing that it's pulling. Is that okay? And the effect that has is it pulls our spindle poles further apart when like we're in anaphase B or we're getting ready for cytokinesis. EG5 is another one that I would know. Again, it's in the kinesin 5 family of plus end directed motors. And it is also going to help to move our spindle poles apart because it will sit on our overlapping interpolar microtubules, and it's going to want to move to the ends. Like if my fingertips were the end of a microtubule, it's going to sit anchored to both. And the only way that it can get to the plus end on either side when it's walking in opposite directions is if we move our plus ends closer together. And the effect that that's going to have is pushing the edges of our cell further apart. So are we okay on push-pull? Okay. All right, spindle assembly. Oh, sorry. Close together. Yes, there are, because you remember how we talked about how you need a balance of push or pull, and if you had like a loss of function in one of these, our spindle would collapse. Like we have equal force being applied in both directions. You don't have to know those as specifically by name, because they don't really, they have a structural purpose, right, in providing a counterbalance to the outward pushing forces, but they're not, at least in this unit, going to be helping with anything like anaphase. So it's more important that you know by name, like egg5 and Dianine. Be familiar with your other kinesins, but like these are the ones that I think you would be most likely to have a question on. So does that, okay. All right, so the purpose of the spindle assembly checkpoint, arguably this is our most important checkpoint after maybe the restriction point. We want to make sure that we have proper attachment, and that's basically like the end of it. This is our last chance before we make our daughter cells to make sure that they're getting the right amount of DNA and that we're not going to have like an extra chromosome or a chromosome missing, broken chromosomes, because a lot of those things, again, could lead to cancer, could lead to cell death, and then you've wasted a lot of nutrients and energy on something that wasn't successful. So this is a really tightly regulated process. And this, again, is checking the tension applied on the kinetochores by our kinetochore microtubule attachments. So once we have attachment on all of our kinetochores, that will just apply like a little bit of pulling from the direction of the poles, and we have sensors, presumably, to detect when there's pulling on all of our kinetochores, and that is a signal that like, okay, we are attached, you know, everything is good, we can go into anaphase and start dividing. So I like Mad and Bub. I think they have fun names. Mad and Bub are our sensors. Again, we don't have this mechanism fully characterized, but they are involved. And once we sense tension, we're going to release CDC20, which is different from CDC6 that we mentioned earlier, that gets phosphorylated when we're preventing origin refiring. If you think that that's going to confuse you, I would just make a note somewhere, maybe on your note sheet, that CDC6 is like beginning of S phase and CDC20 is at the spindle assembly checkpoint. Some of those are confusing when like the name is the same, but the number is different. So CDC20 is released. It's not shown in this diagram, but it's kind of just like an activating protein, and it's going to bind to APC. This APC is APC/C. It stands for anaphase promoting complex, which gives you an idea of like what we're going to be doing. And APC-C is a ubiquitin ligase. So again, we talked about that. Ubiquitin is a little peptide. APC adds ubiquitin to proteins, and that's a signal to the proteasome that like, oh, this is something I need to degrade. So the first thing that we're going to do is we're going to ubiquitinate securin. And securin is, let's see, which one is in this like little gold one up here, and it is holding separase inactive. So when we ubiquitinate securin, then that will be sent to the proteasome, it will be degraded, and then separase will no longer be sequestered. So once we have active separase that isn't being held by securin, it can go and degrade our rings of cohesin. Does somebody want to like remind us what the cohesin rings are, what they're doing? Go ahead. Exactly, yeah. So this is going to be like the beginning of actually allowing our sister chromatids to separate. We're going to degrade our cohesin rings that hold our sister chromatid together, and we are now able to like start pulling from the spindle poles. And this happens by a shortening of the kinetochore microtubules. So this is like, we're moving into anaphase at this point. And also, yeah, APC also will ubiquitinate M-cyclin. So that will be sent to the proteasome, because we can't like proceed through the cell cycle if we still have active M-cyclin. It will still be doing M-phase things, and we need to move to anaphase. So that's kind of a theme. Our cyclins are usually degraded at the end of that phase of the cell cycle, because there are like G1 cyclins, G2 cyclins, S cyclins, and CDK is the one that stays around and is general. Okay, so anaphase. Anaphase A and B. The way that I like to think of the difference between these is everything that involves pulling our chromosomes to either side and like physically separating our chromosomes is going to be anaphase A. And anything that is involved with moving our spindle poles further apart to like create a region that we can cleave during cytokinesis is going to be anaphase B. So anaphase A, we're going to have shortening of our kinetochore microtubules. They basically just get like chewed at the plus end. And as they get chewed, the kinetochore attachment moves closer and closer and closer to the spindle pole. And anaphase B, I think like that's most of what you need to know for anaphase A. Anaphase B is when we have our dynein and our egg 5 starting to push our spindle poles apart. And that just creates like a really clean region right here where, yeah, you can see my mouse. Right here, where we can form our little actin contractile ring and start to squeeze off between our two forming daughter cells and not have to worry about like stuff getting caught in between. Okay, so cytokinesis. Oh, sorry, go ahead. Yeah, there is. Is that kinesin 14? Yeah. Yeah. So kinesin 14, I wish I could reach up there, but kinesin 14 is going to be the one that's sitting like almost at the kinetochore. And as it's attached there, it's going to be chewing back on the plus end. And then like the kinetochore kind of just like keeps re-grabbing at the furthest point along the plus end. So it stays attached. Okay. Yeah, so cytokinesis is pretty straightforward. If any of you have taken anatomy and physiology or physiology, you're probably pretty familiar with actin. It's one of also the 3 cytoskeleton components that we talked about, along with microtubules and intermediate filaments. So we have a little like actin fiber ring that forms in between our 2 cells, and it's almost like an elastic material that's going to start to pinch and squeeze in the middle. And again, we have APC targeting our cyclins, which I think we already talked about. But once we have no more M-cyclin, once we're like not doing M-phase things anymore, this is going to be an indicator for the cell that like it can reform the nuclear envelope because our lamins won't be phosphorylated anymore. Our spindles can break down and we can decondense our chromosomes. So this is like kind of where we're restarting the cycle, and we have 2 daughter cells that are not actively dividing. And that is it for cell cycle 2. So does anybody have questions on that? Okay, then I will give it to John. Oh, sorry.
Right, so also like, there was a dining on the thing or blocking.
Yes. Yeah, that, so, yeah, I probably should have gone into more detail on that. It's gonna be our kinesin 14 that is chewing back on the plus end. And because our dynein is our minus directed motor, that is gonna be the thing that's like kind of following it as it gets chewed back.
Also work that like, they're not attached, right?
What do you mean?
Even like, not every single patch, like, it's still like one.
Do you have an answer for that? I don't know if I'm just not understanding your question. I think it could, but that would be like a mistake, right?
I think like in the video, she showed that like, if it attaches only one side, like it only walks like on one side.
Oh, yes, okay. So yeah, that wouldn't happen under normal healthy conditions, but like say we didn't create our cohesion ring with separates, our dynein would still be strong enough to pull our bound sugar chromatids towards one end. So that's why it's important that like cohesion does get cleaved and we have attachment on both sides. But if we don't have equal attachment, then the dynein will walk back our pairs. Is that what you were asking?
Yeah, that means.
Okay, and that would be like how you have too many chromosomes in one cell, but not enough. So, any other questions on that? That was a good question.
Margot has a question.
There. So. Yeah, probably. There are usually redundant mechanisms that help promote progression through the cell cycle. So a lot of times, even if we don't cover it like in the scope of this class, there might be another protein or another thing that like does a redundant function. But as far as I know, that would probably be like a stuck in the cell cycle. Like if you got an exam question about that, I would say it got stuck in the cell cycle. Just because that kinase won't function with that inhibitory phosphate on it. So we need something to take it off.
Okay. So DNA replication, guys. Let's get started. Okay, so the structure. You guys can hear me okay? Okay. So adenines are gonna bond with thymines, cytosines are gonna bond with guanines, and the phosphodiester bond is what's linking the nucleotides together in the backbone, while hydrogen bonds are being formed in between the two bases. So the strands face each other complementarily, but the way the DNA is structured, you'll have the 5' end on one side and the 3' end on the other side, which gives it anti-parallel structure. Okay, so as I was just talking about, there is a difference in between the hydrogen bond formation between the two types of bases. So A's and T's are going to bond with 2 hydrogen bonds, while C's and G's are going to bond with 3. And we'll look at the structure of the 4 bases in just 1 second. And the helical structure of DNA is going to look, obviously it's a helix, but you'll have major and minor grooves. The minor grooves are just going to be very small, and they occur when the major grooves have that gap. And then the major grooves are just very large in between each other. You can kind of see how it like forms. There's a bigger gap at the bottom, there's a smaller gap at the top. Those are the 2 types of grooves. Is that clear? Yeah, okay. So nucleotide structure, you're gonna have the phosphate, you're gonna have the sugar, and you're gonna have the base. Notice how there's only 1 OH showing on that base, or I mean on that ribose, and that is because of the name deoxy. We're not gonna have 2 hydroxyls coming out of that ribose, only 1 of them. Yes? Yeah. Yeah. Okay. So the nucleotides. So you're gonna have your purines, you're gonna have your pyrimidines. Some people just get it. I'm not one of those people. I had to make a little analogy in my head for so purines are adenine and guanosine. So purines are as pure as gold, that A and that G. And pyrimidines, you can't cut them because of the structure of their cyclic nitrogenous part. So it's not a double cyclic part. And then CUT, uracil is also included, but that's for RNA. And thymine and cytosine are the pyrimidines in this case. And I was talking about this right here. There's no double cyclic structure, so you can't cut them. Charlotte. I don't think,
Never.
yeah, I don't think, yeah.
Like, know that one is hexameric ring. But that's all.
So they're not theoretically supposed to be used interchangeably. So there's guanosine is the actual name for the DNA base. Guanine is so it's the difference between nucleotides and nucleosides. A nucleoside just means it's the triple phosphate version of the base that hasn't been added to DNA yet. So it's a guanosine because it has the 3 phosphates. It's a guanine because it has the 3 phosphates, and it becomes a guanosine once it gets added to the DNA. But for the purpose of this class, we just use them interchangeably. It doesn't matter. Yes. Sequence specific. What do you mean? Oh, the backbone interactions. Yeah. So the major grooves, which are the larger gaps, are sequence specific, and then the minor aren't. Yeah. Okay. Any questions about pyrimidines, purines? No? Okay. So addition nucleotides. So DNA is being read 3 to 5 by the polymerases, and it's being synthesized from 5 to 3. So that means we're adding new nucleotides to the 3' end. And they must, as I just talked about, they must have the 3 phosphates, and polymerase requires that so that you can get the energy from the breakage of from the cleavage of the phosphate bond, so that the base can be added. You don't just spawn energy, like the energy exists, but you just need to take it from somewhere to apply it, for the formation of the bond. So the mechanism, the oxygen is going to attack the first phosphate group, and then that's going to cause the phosphate and the oxygen, the second oxygen, and that's going to cause the breakage of the bond between that second oxygen, which is in blue down here, and the phosphate to break. And then that causes the formation of the phosphodiester bond on the backbone. Any questions about that? Do you know?
Yeah, I do, but I don't think they're. I would ignore it for now, no.
Okay. So we'll get to that in just 1 second, but I'm gonna move forward until we find that out. Okay, and then this is just the type of model. Semi-conservative, all that means is that you're not replicating from one molecule and going straight to the other. You're splitting it and you're replicating to 2 daughter strands, and then that process continues along. What that means is that ultimately, upon the second round of replication, you're gonna end up with 75% of newly synthesized DNA and 25% of old DNA from the template, from the original template strands. So as you can see, first round to second round, 50-50, and then after that. Yeah, go ahead.
It's. Okay, yeah, deoxynucleotide triphosphate, so it's just that other element.
It's just because in this drawing, there's no specific base. Okay, and then this is just an Amoeba Sisters video that you guys can watch later. But it's for replication. Okay, and then for origins of replication, so as we talked about, or as Lily talked about, and we talked about yesterday as well, replication starts in S phase, and you can have multiple different replication bubbles starting. Think about the size of the genome, especially like for humans. You need a lot of these so that it can be done in a timely manner. You're not originating from just one spot and hoping to do the entire genome, because that's just impossible. So you can have multiple at the same time, and they can't, they might fire simultaneously, but not at the same one. So the same, or obviously because they're 2 different origins of replication. Typically, if a region of the genome needs to be expressed faster, you'll have more replication bubbles, and that just makes the process easier. But this is just to say that you have multiple, and for each replication bubble, you're going on both directions, and that just also makes the process faster. Any questions? Okay. So from pre-RC to pre-IC, so the pre-IC is going to be firing in both directions. This is going to happen after ORC already recruited CDC6 and CDT1, and after those 2 recruit the helicase. And the bottom right is a visual of what the replication fork looks like, and you have a couple of key components for this to occur, and we'll go over that next slide. Okay, so you have a whole variety of different proteins that act together so that the process can go smoothly. Important here, for every one of the lifesaver looking clamps, you have a clamp loader protein. They're just not pictured. So you have the important ones that you talked about. We have all the ones coming from the pre-IC. So you have the ORC, you have CDC6, we have ORC, you have CDC6, you have CDT1, and you have the MCM right here. And then you also have the clamp, you have the polymerase, and then you have the single stranded DNA binding protein right here. As we were talking about in class, single stranded DNA is very, very volatile for reactive oxygen species and just other complications about it not being, or not liking to be in single strand form. And so you just have all of those proteins binding to ensure that it remains stable. And so all of these play their own individual roles. So the clamp keeps the polymerase in check. The helicase is split into DNA, and this is all happening really, really fast. And it just goes to show how complex replication can really be. Any questions for this? No? Okay. So just a review, helicase is an ATPase, and it pulls the DNA strands apart. Polymerases are the ones that are adding on to the DNA strand and that are causing the synthesis of DNA. The slide and clamp loader, as I was talking about, is an ATPase that's sliding the sliding clamp on and off the DNA. And it just looks like a lifesaver. I think, do we have like a visual on the movies folder about how that works? It just basically it's wrapped around the DNA and it like gets enclosed in. It's like not really. Yeah. Yeah. And so the clamp is there to make sure that the polymerase is in the right track and that it doesn't slide off. And then single strand DNA binding protein is just binding to the single strand of DNA so that it doesn't get attacked / remain stable. And then the primase is the enzyme that adds the primers as needed for the lagging strand and also once for the leading strand. So lagging strands, leading strands, I have a little tip for this. So if you're asked in the exam, oh, draw a replication bubble, tell me which direction the replication force going on, tell me which one's lagging, which one's leading. So if you're drawing the empty bubble and then you're setting 5, 3, 5, and 3, first off, the 3s and the 5s have to be opposite to each other on either side. But the side that's replicating towards the 5' on either side, so you're divided in half and it's opposite because it's anti-parallel. So the sides going towards the 5 on both ways will be the leading strands. So they're upside down from each other. But if the 5 is here and the 5 is here, where I'm pointing with my hands, and the 3s and 3s are here, the direction that's going, or the strand that has the 5' on the top and in this case on the bottom, are the leading strands. And then the lagging strands are the ones that are going towards the 3. Was that confusing? Okay. Well, this has been asked in the past. When me and Lily took the class, that was actually one of the questions on our final. So it's fair game to just write that on your note sheet. Yeah, we did have to draw it. Yeah. Yeah. But it's fair game. And the lagging strand is called lagging because it's going on the opposite direction of the replication fork. And you have the Okazaki fragments that need to be ligated after the process of replication is done. And that's why it takes longer. Yes. Like total at the end? Yes. Okay, any other questions? No? Okay. So we come to a problem known as the end replication problem. So once the primers get removed and the DNA polymerase is able to fill it in, every single part of the genome gets ligated together besides the very, very end of the 3' end. So why does that happen? Well, it happens because you can't go past it to come back and fill in the gap. And then you end up being in a situation where you just have no way to fix the overhang at the bottom. So once your primers are removed away, DNA polymerase does not have the ability, it cannot synthesize de novo. Is that yeah, it can't synthesize de novo. And so you just do not have the ability to do it by itself. And what ends up happening is that every round of replication, you would end up losing genes because that's just not being filled in. So To fix that problem. Yes. Yep. So to fix that problem, evolutionarily, the DNA has just been extended with repeating non-coding regions to prevent important genes from being chewed off after every round of replication, and they're called the telomeres. And in humans, they're literally just like TTA, GGG repeated like infinite times, so that every round of replication you're not losing anything important, and they're expandable. And also, it just those telomeres are located at the end of the chromosome, and funny meme, I guess, but that visual representation of pink is just the region of the chromosomes that don't really have any sort of coding proteins. They're just there to be chewed off in case, not in case, but over time, they will eventually be chewed off. And it's hypothesized, this isn't going to be asked during the exam, but it's hypothesized that telomeres being chewed off are like what causes aging in organisms. Okay, so to fix that, or to fix the end replication problem, there's this very, very beneficial enzyme called telomerase. And so what it does is it carries its own RNA sequence, and it's going to go on to the end of that 5' gap, and it has complementarity to the end sequence. So what it does is it goes on with its own complementary sequence, and it binds, and it extends that it extends the end part of the 3' end so that a polymerase can go in and fill in the necessary bases. So it's going on, it's binding complementarity. I mean, complementarily to the 5' gap, and it's extending that 5' gap so that polymerase can go in and fill in the missing bases. I should have been moving with the mouse. Okay, so it's binding at the end of the 3'. It's a 5' gap right here because it's the 5' for the bottom strand. It's going on, it's complementarily binding right here. It's extending this part, and then the polymerase is going on. Yes.
It has full RNA, but it's still in DNA. Telomerase is an RNA-directed DNA. I didn't know that.
Yeah. And so that allows for polymerase to have a primer to bind and for it to fill in that last 5' gap. But what ends up happening is every time this is done, you're losing parts of the telomere. Right? Yeah.
And then it's with,
It doesn't get fixed. The telomeres just shorten.
oh yeah, but there's a way of altering that. Yeah. Like it's kind of like in like an add basis area.
That's it. But it doesn't fix the well, it's a patch for the problem, but the problem continues to happen.
Oh, gotcha.
So the telomeres get shortened over time. So that's the end for topic 9. If we have questions about, yes?
Yeah.
It lengthens. So it lengthens the 3' end. That's it's not complementary to the 5' gap, but it's where the primer was last removed on the 3' strand, so that what you don't have is that final part on the 5' end, because there's no upstream basis for you to add the polymerase in, and it can't just randomly go on and synthesize. It needs a primer. So what the telomerase is doing is it's adding with its own complementary RNA sequence a primer for the polymerase to go onto. Well, technically, yes, because it's complementary to the end of the telomere. But the telomere is just a non-coding repeating DNA sequence at the end of the chromosome.
But the bases that it's filling in are.
Yeah. Yes. It patches it, but over time the telomeres just get shortened because of your non-repeating sequence. I mean your repeating sequence. You'll have a 5' gap every time, and you're extending it a little bit and fixing the remaining like gap. But this happens at every single round of replication. And so over time, do you have a better explanation for that?
So I think telomerase, when it's filling in, like it will mostly solve the end replication problem. The thing is, we don't always necessarily want to solve it, because as we replicate a chromosome more and more times, our telomere, because of the end replication problem, is gonna have that overhanging edge. Our overhanging edge is gonna get clipped off, and so they're gonna get shorter and shorter and shorter. That is an indication of aging, and an aged chromosome is one that is divided more times and is more likely to accumulate mutations. So when our telomeres shorten, that's a good indicator for the cell, like, okay, we need to recycle this, we need to not use this anymore, this happens for cancer. So you see these a lot in some other organisms of developing embryos of fetuses, you see these. But normally we just don't want to solve the end replication. Like, it's a good thing to know how old our chromosomes are. So it can solve it, but that's not always in the best interest of the organism. Usually you just want them to get clipped off progressively. Does that answer your question? Yeah.
Any more questions before we move forward? Yes. So the added polymerase bases, so the added telomerase bases act as a primer for the polymerase, and then the same, it's basically patching in between Okazaki fragments, but at the end.
Basically, like, since the DNA polymerase is unidirectional in the way that it can synthesize DNA, we have this lagging end, right? So we didn't have telomerase. You can't synthesize this way. It can only go this way. So telomerase is giving us a little sequence here that we can backfill in going the way that DNA polymerase will want to work. That's like basically it. If that, like,
The telomerase is where DNA polymerase or not telomerase, the telomeres where
The bases added to the end of the telomeres by the telomerase act as a primer for polymerase to go in and bind.
so telomerase adds primer.
Yes, but it's not an RNA primer. It's a DNA primer. Yes.
So primer that's added, that's the telomere.
The telomere is the end region of chromosomes, and at every end of a 5' part of a telomere, you won't have. So because the primers are removed at the end of replication, or I mean at the end of replication, you're going to have an overhang at the end, right there at the 5'. So the 3' end is filled all the way to the brim, but because polymerase can't synthesize de novo, so it can't just like add without primer, it can't lodge in, and so it just can't polymerize on the right direction. And so telomerase goes in at the end, extends that region so that polymerase can go on and bind and finish filling the gap. Yeah, so it's like a multiple thousand base pairs that are repeating and non-coding. Yeah.
What telomerase is not expressed in all types of cells, so is that like? Is it not always expressed so that you can like see the aging, like you're saying?
Yeah,
Like
a
you
lot
don't
of
want it.
times, like, it's just not detrimental enough to even, like, in an episode, right? You're gonna be dividing, I don't even know how many times, and so it's really important that like your chromosomes aren't aging out after a month. But for us, most of our cells at any given point are not dividing, so it's better to be able to see how old they are than like to waste all of this energy on expressing telomerase and like fixing this end replication problem and potentially using the fail-safe that would protect us.
So it would just like, it would just keep getting shorter.
Yeah, so you can think of the telomeres as like a buffer. Like we have a certain allowance that you can start chewing those back before you're gonna get into the coding region and normally like your natural lifespan is not that long.
It's like a potato for Plants vs Zombies. No, like unironic, that's how I like studied it for this class. Okay. So we're gonna move on to topic 10. Let me just check the chat.
Still have questions about that at the end. I know it's fun to take them off.
Yeah.
So, with the telomerase.
Mhm.
Would this be happening after the leading strand is finished replicating or during leading strand replication?
So you know how, like, for you to complete replication, let's move farther into the chromosome, not at the edge. So you know how when you have the Okazaki fragments,
Yeah.
the primers get removed so that DNA polymerase can go back in and finish. You can't do that at the end because you can't go farther upstream. And so it's happening, it only happens once leading strand synthesis is done.
Okay, I guess because the replosome can't like.
It can't go, so how you fix the gaps in between the Okazaki fragments is you go post Okazaki fragment gap and you fill back in, right?
As in after all the Okazaki fragments?
No, each individual one. So each individual one will have more machinery so that polymerase can rebind and then refill. And then that can't happen at the end because you can't go farther upstream until something like telomerase goes in and adds a primer. Okay. With telomerase, I want to make sure I understand this. Templates with it's made of yeah, so the telomerase template is made of RNA and it's acting as a reverse transcriptase to add more DNA bases to the end. So it's added to the end 3' end. And so that acts as a primer so polymerase can go in and bind and fill that region. Yes, so it's backwards from the RNA primer.
Telomerase is only functioning at the actual end.
At the actual
So
end,
it's not between.
not in the middle of the Okazaki fragments.
The normal Okazaki fragments is filled in by ligand.
Yes. Yeah, of course. Okay. Oh, topic 10. Okay, you guys got lucky. It got shortened this semester.
Is it really bad in other semesters?
No, it's very interesting, but like my favorite concepts for topic 10 got like chopped off. BER, HDR, and HEJ, like all of it, yeah. Yeah. Okay. So polymerase proofreading. So if there's no polymerase proofreading, you'd have about an error from 10,000 to 100,000 base pairs being added. It's not a really good number considering how detrimental to gene expression that can be. And so polymerase has a proofreading mechanism that lowers that to about 1 into every 10 million base pairs. And so once DNA is being added, or I mean, once DNA is being synthesized by polymerase, and it senses an error, what it does is it shifts that base onto its exonuclease domain, and then that gets excised. And then once that happens, polymerase goes back in with its polymerizing domain and adds the correct nucleotide or nucleotide. So the exonuclease has 3 to 5 activity, and so it binds the 3' end, like I was just saying, and it chews it back. And then after that's done, it just goes back into the polymerase site.
Does the polymerase like flip over?
Yes.
Okay.
So the drawing right there, it's synthesizing normally, it senses a mistake, it sends the base pair onto its exonuclease site, chews back on it, and then fills in the right primer. I mean, the right nucleotide. And polymerase theoretically looks like a right hand like this, and the sites are at the palm. Okay, so mismatch repair. It's a process that happens post DNA replication and post polymerase proofreading. And it's just to catch any sort of errors that polymerase does not catch. And And so it must be able to tell which one is the parent strand and which strand has just been recently replicated. So in eukaryotes, you'll see this as nicks in the DNA that don't get quite patched just yet. And in prokaryotes, you'll see methylated DNA. And MMR is able to tell how nucleotides aren't supposed to be there because the nucleotides, oh crap, where is it? It's later on. Okay, so it's able to tell which DNA has just been misplaced by telling which one's the parent strand and by sensing a lesion in the DNA. So because they need to complementary pair, if you have the wrong nucleotide, you'll have a little groove on the backbone, and I'll show that in just 1 second, and there's a slide that shows it. And MMR must be able to distinguish which one to fix. And yeah. So the process involves being able to determine which one is, or which strand is which. And once a mismatch is sensed, as Laura was talking about in class, you're gonna remove a whole lot of nucleotides just to fix that one mismatch. So it's a process that involves a lot of resources and a lot of energy. So it's preferable that polymerase just catches its own mistakes. But it exists in order to prevent large single nucleotide errors. Yes.
Can you just go back or which one is unmethylated, where it's methylated again?
Methylated will have the newly synthesized strand will be methylated for prokaryotes. The new strand is okay, reverse. So the new strand is not methylated.
Sure. So prokaryotic DNA is methylated by an enzyme. I don't know what it's called, but there's one that adds methyl groups along the backbone. And that happens after the strand is synthesized. So when you have a newly synthesized strand, it won't have any methyl groups on it yet. And so that's how, like, you can tell when you're proofreading which one is the new strand that has been mistaken, because it won't be methylated. But within like 30 minutes, that enzyme will go through and methylate the new strand, and then you won't be able to tell which one's the one that's old. So you have to correct your errors within that strand. Otherwise, it's like a 50-50 shot. If both are methylated, you can't tell, and you might make an edit on the parent one by mistake and then have a mutation on both strands.
Thank you.
Okay. So once that happens, you'll see a little bubble form. And then the large groove of a lot of nucleotides will also be encompassed by the cell machinery. And so helicase is going to separate both strands, and then an exonuclease is going to come in and chew back on the newly synthesized strand, theoretically, if MMR picks it right. And then sliding clamp will be added by the clamp loader, and then polymerase will be added on, and it'll fill back the missing nucleotides. And then after that, the ligase can go in for eukaryotes and seal that nick. Any questions? Okay, so this is just the visual representation I was telling you about, but the lesion, and there can occur incorrect identification. As we were saying, and there was a problem on topic 10 problem set. That was like a hypothetical, and I wanted to explain that. So if no repair occurs, then the mismatch pair continues, and you'll have one daughter cell with the correct nucleotides and one daughter cell with the mutation. If repair occurs on the parental strand, you'll have both daughters with the mutation because you're taking out the wrong nucleotide. You're taking out the correct, or not the wrong nucleotide. You're taking out the original nucleotide and keeping the mutated one in. And if repair occurs on newly synthesized strand, then you're going to have the MR process occur properly. So the question, just give me 1 second. So the question on the problem set involved asking, oh, if I had a cell that did MMR, but could not distinguish which one was the newly synthesized and which one was the parent strand, and it is doing MMR, and then you had one that wasn't doing MMR at all, how will the error rates compare to each other? And so basically, if you are doing MMR, but you can't tell which one is which, 50% of the time, you'll have a cell that will end up with the mutation, and 50% of the time, you won't have a cell that will end up with the mutation. And then if you weren't to do MMR at all, you'll end up with a 50-50 shot of you having a mutation or not having a mutation. So in the end, they'll just compare equally. So if you're checking for, if you're proofreading for MMR, but you can't tell which one's which, the odds are that half of the time you'll fix the correct mutation and half of the time you won't fix the right nucleotide. And then once further replication happens, you'll have an entire line of cells with that mutation and one entire line of daughter cells without that mutation. And that's why the error rates with no MMR and MMR like that are the same. Yes. Mm-hmm. So for replication to occur, you're splitting the strands, right? So if you're replicating the entire bottom strand and it has the correct guanine, forever on you'll have the correct guanine and the complementary cytosine. For the top strand, because you have the adenine and you're replicating the top strand and doesn't get caught because there's no MMR, the entire line of daughter cells with that was used with that template strand now, because it didn't distinguish, will have the mutated sequence. Yes. I have no idea. Yes.
So if for example you had like a prokaryote and like neither strand was
Methylated.
then like the MMR protein would be able to differentiate. But if both strands were methylated,
Same case.
so it'll still like attack a methylated strand.
It'll still do 50-50. Yes.
So the reason that LMR
Yeah, it still senses, so it's because you're not changing it, so it'll remain like that. And well, if the polymerase missed it and there's no mismatch, for cases where we're studying just mismatch repair, it'll remain like that. There are other nucleotide like exchange mechanisms that happen for DNA proofreading that we just didn't cover this semester. Yes. So the question for the problem set asked if there was an overly active enzyme that methylated both strands. The reason why so if it senses the methyl on one end, it'll fix the other, correct? So if you have methyls on both, it's a 50-50 shot of whichever strand will be picked, the other one will be mutated. Yeah, so it does on the opposite, but it sees the methyl on the backbone, and so it just slides onto the other. Yeah. Yeah.
For that question, either way, if it's under methylated or
It's the same result. Damn. Okay. So that'll be all for today's review. That's one of our old TA's memes. I miss Gabe. But yeah, so feel free to email us with any questions. And I hope the reviews were helpful. Laura is hosting a review session in her office, or not a review session, it's like a Q&A from 5 to 7 tonight. So if you have last minute questions that you want to ask her, feel free to do so. Let me just stop the recording.
If I can find it, oh.